coming to the Computational Science Research Colloquium series. Today, I have the pleasure to welcome Sanford Bernstein, who's gonna tell us everything about muscle and flies. Take it away, Bernie. Sandy.
All right, well, that was short but sweet, and I'll put up my slides here. Let's see. Are you seeing that, Jose?
Yes, you can see fine.
Do you just see the slide itself? Because
We see the slide and see the cursor also.
The thumbnails also are showing up. Sandy.
Yeah, that's what I'm going to try to get rid of here.
Oh, it's okay. We can read ahead.
Let me see. So check again here. Not sure why it's doing this, Jose. It didn't do it before. And we won't be able to see the movies if I do that. Oh, I know. There we go. Better?
Yeah.
Okay. So going to talk about our work, which is on Drosophila. Well, of course, very sorry here, but we seem to have skipped way down to the end. Isn't Zoom fun?
Till you have too much.
Yeah. Okay. So we're going to talk about using flies to understand heart disease, specifically disease that's involved with the protein myosin, myosin heavy chain. So here's a little primer about muscle. First, we'll talk about skeletal muscle, shown here. It contains a bundle of muscle fibers, cells that are multinucleate in nature. And within each cell, there are these subunits called myofibrils. And the myofibrils are made up of repeating units called sarcomeres. And those are what are responsible, among other things, for contraction of the muscle. And the way that works, if we look at a diagrammatic version of the sarcomere over here, there are two main components. The thick filament and the thin filament, okay? And the thick filament is composed of largely of myosin, which is the protein we'll focus on, and the thin filament composed largely of actin. And in both a calcium and ATP dependent fashion, when the muscle is stimulated, the myosin heads grab on to the actin thin filaments and walk along them so that the muscle can contract like so, okay? The sarcomere contracts, the whole myofibril contracts, and as a result, the muscle fiber contracts. Okay, so this is more detail than you need to know, but I'll just briefly show it to you because it also introduces you to the myosin protein structure. As I said, there's a head of myosin that interacts with the purple bar, and that's the actin filament, okay? And in the presence, so here's the globular head. It has a actin binding site. It has an ATP binding site, and it's the hydrolysis of the ATP that's important for movement, okay, energy, that serves as the energy source. So in addition to the motor domain, the globular head, there's this long alpha helix, which has a light chain attached to it, and that intertwines with another alpha helix and many other myosin molecules to form the thick filament way down here. So when ATP comes in, the myosin detaches from actin, and you get so-called recovery stroke that's sort of concomitant with ATP hydrolysis, where you actually get movement. This is the molecular motor portion of it. You get movement of this long so-called lever arm relative to the head, okay? Then you get attachment to the actin, again, because it's a conformational change. And what occurs is that the stored energy is released and you get movement of this lever arm again, and that's called the power stroke. And that power stroke actually ratchets up the actin filament relative to the myosin. Then the phosphate comes off, then the ADP comes off, and you come in again to the cycle, so forth. So basically you get movement as a result of hydrolysis of ATP by the myosin, and some of the key areas are the actin binding site, the ATP binding and hydrolysis site, and this lever arm that serves to allow the power stroke to occur as a result of movement of the arm. Okay, so this is a little video that was put together many years ago by Jeff Sale and Roger Sabadini that shows you graphically the myosin head interacting with the orange thin filament. The ATP is coming on and getting hydrolyzed. There's another protein called tropomyosin that moves out of the way so the myosin can interact with the actin. Okay, so myosin binds the thin filament, uses ATP to change shape and move the thin filament. Kind of mesmerizing, I think. So from our angle, we're interested in the basic function of the myosin protein, but also in mutations in myosin that cause human disease. And we've studied a number of skeletal muscle diseases that are a result of defects in the myosin molecule. But, and I'll briefly mention those, but we'll also talk about hypertrophic and dilated cardiomyopathies, which are relatively common inherited diseases that are cause severe effects in humans. Okay, so what our lab has developed over these 37 years that I've been here is an integrative analysis of myosin isoform function. So what do I mean by this? Well, we use fruit flies as our model system, and we express mutated versions of the myosin and make transgenic fruit flies that express, instead of the normal form of myosin in their muscles, they express a mutant form. It can be a mutant that we're interested in studying for basic science, or it could be a model of the human disease that we study, okay? So in terms of the integrative analysis, once we express it in the fruit fly, we're actually able to isolate the protein and look at its structure. A lot of this with Tom Huxford in chemistry by crystallography. We've also negatively stained, and we're starting to do cryo-EM for structural analysis. Once the protein's isolated, we could also do biochemistry, transient with collaborator Mike Jeeves, and steady state ATPase assays, as well as actin binding assays. We can do biophysical level, which one assay we do is the in vitro motility assay, where we actually watch fluorescent actin filaments move along a cover slip that's coated with myosin and measure the velocity of the movement. So we could see that some myosins will cause reduced movement of the actin, and some actually will increase it. And optical trapping that I won't talk about, but that can measure force output and how much the myosin head moves. Then we move up to the cell level, and we do a lot of confocal and particularly CRYO-ELECTRON microscopy to understand the structural defects that occur in the muscles themselves as a result of the mutations. And then we worked with my former postdoc at Rensselaer Polytech, Doug Swank, to do skeletal muscle mechanics, where we could measure various parameters. I'll talk a little about that: force, stiffness, work, and power. And more recently, we've moved into heart physiology that I'll talk about, where we can measure the actual fruit fly heart that's containing this defective myosin and see how it affects parameters such as heartbeat, the diameter of the heart, fractional shortening. And then finally, we can do the whole organism locomotion, measuring flight ability of the flies, for instance, wing beat frequency. Okay. So this is a fairly complicated molecular biology story that we spent many years on, but I'm just going to very briefly go over it to give you a handle on the myosin gene. I know we have a diverse audience, all the way from physicists to MDs. So basically what this is showing is the structure of the myosin heavy chain gene in the fruit fly, Drosophila melanogaster. Okay, here's the adult and there's the embryonic stage, and what's in green are the various muscles. And they do have many different muscles, even though they're very simple organisms, in quote. Flight muscle, abdominal body wall, leg muscle, larval body wall muscle, pharyngeal muscle, proboscis muscle, et cetera, et cetera. And the way they handle these different muscles is that they make many different forms, as many as 21 different forms of the myosin protein. And the way they do this is not by using different genes, it's by using a single gene and using a process called alternative RNA splicing. So you may remember, you start with the gene, you make the RNA transcript, that gets processed, and that's codes for the protein. Well, there's a complex processing mechanism for myosin where the coding region from the transcript from exon 1 and 2 are included, but then the processing either chooses 3A or 3B, then 4, 5, and 6, and then 7A or B or C or D, et cetera, down the line, so that the final outcome of the RNA and protein could be 480 different combinations of types of myosins that are produced. We, as I said, know about 21 different forms. Okay, so we've spent a lot of time studying the different forms and trying to understand what the different domains that are encoded by these different alternative exons do. And in fact, here's the structure that we worked on with Ron Milligan at Scripps. This is the chicken myosin, but it's similar to Drosophila, as you'll see in a while. And here's the globular head, and here's the long lever arm, and here are the two light chains that bind. And then we have all these different exon alternative regions, and they're in kind of important areas for the protein. Like this one is near the ATP binding site. This one might interact with the actin binding site, and then down to this converter region, which interacts with the lever arm. So you could see that if you change the amino acid structure of those particular alternative regions, you might change the functional properties of the protein as a result. And that's something that we spent a lot of time testing, and I'm just going over in a thumbnail sketch. But basically what we do is express the wrong form of myosin in the flight muscle. We use the flight muscle, the indirect flight muscle, as our manufacturer, and we have a mutant that knocks out the normal myosin, and we express, for instance, the embryonic myosin in the flight muscle. And then we can do all those, and I should point out, all the alternative domains in the embryonic are different from the flight muscle. So we can see how that affects the structure of the muscle, the function of the myosin, et cetera, the ability to fly. And then we could take it a step further, and we could swap in one alternative exon from here into the embryonic, take one alternative domain from the embryonic, put in the flight muscle. So you can see there are a lot of combinations we could do, and we've published quite a lot of papers on that, and just came up with a few tidbits here on this last slide about this. So this region near the N-terminus seems to affect the ATPase the in vitro motility, the fiber power, and the flight ability of the fly. The region near the nucleotide binding site affects the ADP release rate and the power ability of the fibers. This region called the relay domain affects fiber power, load sensitivity, and flight. The converter domain affects in vitro motility and flight, and then two regions outside the head, the hinge, affect the sarcomere length and flight, and the C-terminus involves myofibril stability. So a complex story that I'm going over in a flash here, but at least it introduces you to the myosin protein and the various domains and the differences that occur in different isoforms of the protein. Now, I'll talk a little bit about the structural studies. This just came out in Journal of Molecular Biology. It's a collaborative study with Tom Huxford's lab. Now, we were interested in isolating the Drosophila protein and understanding its structure at the molecular level, at the atomic level. The problem with Drosophila, the fruit fly, it's a great genetic system, but it is very, very tiny, right? So we dissect out 100 flight muscles and get enough to do the ATPase assays, but we couldn't get enough to do crystallography. So what we decided to do was use our fruit fly as a manufacturing system, and we created in order to get enough myosin for structural studies. And what we did was hook up a transcriptional promoter that allowed expression, in this case, of the embryonic form of myosin, specifically in the flight muscle. But we made it special. We also added, you could hardly see it probably, this HT, a little his tag, okay? His tagged embryonic indirect flight muscle expression vector. We made transgenic flies. They're expressing the wrong form with a his tag in their flight muscles. They couldn't fly. They have the wrong form of myosin that doesn't function appropriately, but it accumulated. Then instead of dissecting out 100 to 200 indirect flight muscles, what James developed was this, what he calls a fly slushy, where he would take 10,000 flies and grind them up. And now you have every form of myosin that's being expressed in this fly, in addition to the his tag myosin. And if you know the biochemical trick that you can run a chromatograph, we do a myosin isolation, the protein is up here, myosin heavy chain, and then we run it through a nickel ion column, and we're able to specifically isolate the his tagged version. All the other myosins and all the other proteins flowed through, and then we could treat with proteolysis, chymotrypsin, and isolate the globular head and get pure single form of S1. And James was successful in crystallizing this protein, took it to the national beamline, got the fraction to 2.2 angstroms, and he and Tom were able to solve the first insect myosin structure. Okay, and as I said, we just published this, and tell you the truth, it didn't show anything amazingly surprising, which in some regards is good because it's very similar in structure to the human protein. So it suggests that making the mutants in the fly model is a reasonable way to go. Okay, we mapped where the alternative domains encoded by the alternative exons are. Okay, and we did also work with a group at UCSD to do molecular dynamics. And we modeled the flight muscle form computationally and compared it to the embryonic form that we had the crystal structure of. And what we found was that the flight muscle form showed increased mobility in a couple of areas that suggest a reason, both here where the lever arm is and over here where the actin binding site is, suggesting that perhaps this increased mobility that's present in the flight muscle form, according to the molecular dynamics, might account for the ability of this to be a very fast myosin, because we know it hydrolyzes ATP much more rapidly. We know it moves actin much more readily. And we know, of course, that it works in this super fast flight muscle that can beat hundreds of beats per second. Okay. So where we are now on this story is that we've also actually determined the structure of the indirect flight muscle isoform. And Tom is busy at work comparing the details of the alternative domains of the flight muscle form versus the embryonic form in the hope of really understanding the structure-function relationship, where the differences are encoded by those particular amino acids that differ between the two. So we shall see how that story develops. So now I'm finally going to talk about the topic of the seminar, at least the title of the seminar, which is heart disease. As I said, both dilated cardiomyopathy and hypertrophic cardiomyopathy can be caused by mutations in this protein myosin. I think the hypertrophic ones are shown as blue dots, and some of the dilated forms are shown as red dots. Hypertrophic cardiomyopathy, you get increased mass of the left ventricle. And dilated cardiomyopathy, you have very poor pumping and thinning of the cardiac wall, and that's a very severe disease. Okay. So just focus on two proteins, two mutants that we made in the fly to try and understand the basis of the disease. The first one, it's a human mutation or cognate of the human mutation, R146N. And interestingly, what we found is when we did the modeling of the structure is that that region is actually in the post-power stroke state adjacent to another amino acid or a couple of them, and they interact, and that interaction is in this long lever arm. So you might think that it's kind of stabilizing this interaction because there's this charge interaction from the mutant residue to the wild type, to the lever arm. Okay. But in the mutation, what we found was that it kind of destroyed that interaction. Okay. So you might expect that as a result of the mutation, you're going to destabilize the lever arm main motor interaction. Okay, that's just a hypothesis. Okay. But you might think, well, let's see, what's that going to do? Maybe that means that it's going to go through the cycle more rapidly because it's not stabilized in this post-power stroke state. And in fact, that's what we found, that the mutation enhances at least the basal ATPase activity. So this is a ATPase graph where we determine how rapidly ATP was hydrolyzed. And what we see, for instance, here for the magnesium ATPase, there's a dramatic increase in the ability here to occur, to hydrolyze ATP. So in fact, this really correlates what's being found in the mouse models and the human situation. These hypertrophic cardiomyopathy mutations seem to enhance the muscle function, the cardiac muscle function, okay? And the heart has a lot of trouble dealing with this enhanced function, and it seems to increase so-called hypertrophy as a result of that, and gets out of whack in terms of its pumping capability. So what happens when we express it in the flight muscle? Okay, so now we're looking at the sarcomeres of a normal flight muscle over aging. What do we have? Pupal stage, 2 days, 2 hours, and 2 weeks, I think. And here's the sarcomeres, like I showed you before. And what we're seeing up here, these are electron micrographs of cross sections of the sarcomeres, where you have those thick and thin filaments interdigitating. So this is the normal muscle of the flight muscle. And then when we express the R146N mutation, it assembles normally, but within a couple of days, there seems to be some problems in the structure shown by the arrows. And later on, a couple of weeks, there's a lot of disarray when you look at this sarcomere, of course, in longitudinal section. So it appears that, you know, somewhat akin to the human situation, we are getting disarray as a result of expressing one of these hypertrophic cardiomyopathy myosin mutations in the fly. So flies do have hearts. People don't really know too much about that. People wonder whether they have muscles, but I think you know that, but maybe you don't know that they have hearts. And it's this long tube in the abdomen. It's an open circulatory system. And our collaborators, Karen Oker and Rolf Bodmer at Sanford Bernstein developed a system where we could actually look at the fly heart as it's pumping and try and understand the structural and functional defects that occur in this pumping heart, and even in a computational way, since this is the computational sciences seminar. Okay, so what they do is, and Tony Camarado and Gadish Melkani and Adriana Trujillo have been doing this at our lab and at our collaborators' lab, Tony's lab and Johns Hopkins, cut open the fly's abdomen, and it's on its back, and you remove the guts, and you're able to see the beating heart. You cut the neural inputs and put some saline in, and then you do a video imaging of the beating heart. So you can see that heart beating. And then you take a single pixel slice through the heart over time, over a few seconds, and you splay that out as a chymogram. And from that, you can determine all these functional parameters: heart rate, heart period, systolic and diastolic intervals when it's open and closed, fractional shortening, the percent of shortening, whether there's fibrillation, arrhythmicity, and so forth. Okay, so these are not the mutants that I'm talking about today, but they're a pretty good demonstration of what happens. So here's our heart. Here it is stained for the actin molecule. And here's the wild type heart beating in the video microscopy. Okay. And when we looked at the hypertrophic cardiomyopathy heart, it looked akin to this. So if you look closely, you can see that it's actually narrower than the normal form. And as I'll talk about in a minute, we also have a dilated cardiomyopathy-like model where the heart is actually wider than the control. Okay. And in fact, we can see that here when we look at the chymogram, diastole, systole, diastole, systole, diastole, systole. So you could measure that distance over a number of diastoles, number of systoles, compare it to the mutants. And this one is the dilated one, this is the restricted one. So what I mean by restricted, we think what's happening is that the myosin is overactive, and as a result, the heart muscle stays contracted more. Okay. So it doesn't seem to be enlarging like the case in the human situation, but there is a human disease called restrictive cardiomyopathy that has some similarities to hypertrophic cardiomyopathy, so this might be a similar story. Adriana did some electron micrographs of the control and the mutant of the restricted heart. And indeed, we did find that there were a lot of these abnormalities of the myofibril structure that occurred in the mutant. Okay. So here is the data, or are the data, and maybe it's more than you want to know, but compared to the control. Here's the mutant, and you can see the diastolic diameter is reduced and the systolic diameter is reduced, and also the fractional shortening is reduced. Okay. And these are the chymograms. You can see here's the control, very wide. Here's the mutant, narrowed. And actually what our collaborators did at Hopkins was try and figure out what's going on here. They treated these hearts with chemical EGTA that removes calcium that's important for heart contraction or muscle contraction. And when they did that, they saw that there was an increase of the diameter of the mutant heart dramatically compared to the control, wild type. And then further treatment with this chemical inhibitor of myosin function even more dramatically reduced, showed a heart size increase. So in other words, you were able to remove this restricted heart if you prevented the myosin-actin interaction. Okay, so that went along with the theory that there probably does seem to be an excessive cross-bridge interaction occurring in the mutant. Okay, so that was the hypertrophic cardiomyopathy story. The dilated cardiomyopathy story, Adriana just defended her thesis on this, dissertation on this. The mutation we studied was S532P. It's in the location where it interacts, or thought to interact, with actin. So it's supposedly at the actin binding site. So she did a number of assays on the isolated protein. She found that it had a somewhat less affinity, but not statistically significant, between the mutant myosin and actin compared to the control. But Karen Su, who did the ATPase assays, found that there was a dramatic reduction in the Vmax of ATPase. Okay, so this seems to be an underfunctional protein in the fruit fly system. Again, very similar to the human and mouse models, suggesting that dilated cardiomyopathy has underfunctional myosin or other proteins involved in muscle contraction. Okay, so I just want to briefly introduce you to this work from Doug Swank's lab, because I know some of you are engineer types and you can talk about this more than I can, but I'll briefly try to explain it. He actually looks at the ability of the muscle fibers in the flight muscle to contract, produce force, power, stiffness, and so forth. And the way he does it, he dissects out individual fibers from these flight muscles, bathes them into a detergent, adds the calcium and ATP, and when they're hooked up to a force transducer and a lengthening, shortening motor, piezo motor. Okay, and as a result, from what I understand, it allows you to determine the elastic and the viscous properties of the muscle as if you're studying some engineering material. And in the presence of ATP, you can determine such things as power output and various aspects of the cross-bridge cycle. Don't ask me to explain it in too much more detail than that, but it did show that there's a dramatic reduction in power output by the mutant flight muscles that have this dilated cardiomyopathy mutation in it compared to the control. And that power peak was at a lower frequency. Okay. The frequency of the flight muscle in vivo is about 200 hertz. So it's kind of cool that the power peak is right where that muscle is tuned to go in the natural situation. Okay, and then for you computational aficionados, a number of parameters were determined from the sinusoidal analysis of pushing and pulling at different frequencies. And what was interesting is that it resulted in a reduction, slower rate or weakened affinity of actin for myosin, and an enhancement of the cross-bridge, so a reduction in the cross-bridge binding ability, and which is the myosin to actin bridge, and an enhancement of the dissociation of the myosin to actin bridge. So that's a faster rate. So basically, weakened affinity between the actin and myosin was shown by this particular assay. Okay. And we also found that flight ability was dramatically reduced, and the reduction increased over time, the red versus the control in blue. But also, we found that there was no effect on the structure of the muscle. Okay. So this was different than the story for hypertrophic cardiomyopathy. So the muscle, as opposed to tearing itself apart, seems to look fairly normal. And you might understand this. It's an underfunctional myosin, so it wouldn't necessarily disrupt the structure of the protein. An overfunctional myosin might. Okay. And then finally, we looked at the contractile dynamics, and make a long story short, and it's a little bit more of a complex story, but basically, at least at 3 weeks, when you compare it to the control, there is dilation of the heart, the diastolic diameter. Okay. So where Adriana's going on this is that we're trying to understand, again, the structure function properties of the myosin. How does that single mutation cause dilated cardiomyopathy? So she made the his-tag version, and she isolated a lot of the protein, and she tried crystallography, many iterations using a robot to produce all these different crystallization conditions. She got crystals, but they never diffracted to appropriate diffraction levels necessary for determining the structure. So we're taking a different tack. We're taking actin filaments, and we're attaching myosin heads, the mutant and the wild type, separately. And this is so-called arrowhead structure, shown in negative staining that she's taken nice pictures of. And working with a group now at Scintillon Institute in town, we're doing cryo-electron microscopy that will allow us to image hundreds and hundreds of these images, put them together computationally, and determine a high resolution structure that can be almost as good as the crystal structure. The advantage in this particular mutant is that since we're dealing with a mutation that's at the actin binding site, it will be cool to see if the actin binding site is disrupted, because we'll actually have the myosin binding to the actin. Okay. So that's work that's in progress at this point. Okay. So to conclude, domains encoded by alternative exons in Drosophila myosin define myosin functional parameters. We can isolate myosin in adequate quantities from Drosophila, from our expression system for crystallography and now for cryo-EM imaging. We argue that Drosophila produces useful models for human myosin cardiomyopathies. Overactive myosin seems to yield this restrictive heart phenotype in flies. Underactive myosin can yield this dilated phenotype, similar to the human situation. And in terms of future work, flies might end up being a good model system. For instance, if you have a human patient that has a particular mutation in myosin, but you don't know that that's necessarily causative of the disease, you could put it into the fly and see if it does cause this restrictive or dilated phenotype. In addition, there's the possibility of developing either genetic or pharmacological therapeutic strategies, for instance, adding drugs to our cardiac system, in vitro system, to see if that changes structure and function. Okay. So I think 3 more slides. I just briefly mentioned that I'm not talking about this, but we've studied a number of other human diseases that cause skeletal muscle phenotypes, and they surely affect fly muscle dramatically. This is the one that causes myosin mutation that causes inclusion body myopathy type 3. We get inclusions and dramatically upset muscle structure. This one causes distal arthrogryposis. It's a rare childhood disease where you get clenched fists, even facial contortions. And we find that the muscles are indeed very stiff in the flies, just like the situation in humans as a result of these myosin mutations. We've studied myosin storage myopathy, which are results from mutations actually in the myosin rod rather than the globular head. And there's problems both with assembly and stability of the myosin filaments. And we've studied restrictive cardiomyopathy mutation as well and found that some of the defects that occur with that. And we're just working now on this mouthful here, spondylocarpotarsal synostosis that we got an R21 last year from NIH to study the defects that occur. That's another disease that's in the embryonic myosins of humans. Okay. So not everyone that's important is here, but a number of the people are. James Caldwell, who did the crystallography studies, Adriana Trujillo, who did the dilated cardiomyopathy studies, Karen Su, who did a number of the ATPase studies, as did Gurish Melkani, Bill Cronert, who just retired from my lab after 37 years. So going to be a little tough without him, but he made most of the mutants that we talked about today and did a large amount of the electron microscopy. And Jennifer Suggs, who's still here, thank goodness, going at 25 years. So she's done a lot of the electron microscopy as well as the genetic studies that we've shown here. And then here are my collaborators. I'll just point out a few that are particularly important to what I talked about today. Fiber mechanics, Doug Swank and Tony Camarado doing the heart work, and Ross Walker at UCSD, now at GSK, and particularly Tom Huxford on the crystallography work. Mike Jeeves, we've done a lot of collaboration on the myosin ATPase studies. And these are our relatively new collaborators, Dorit and Niels, who are doing with Adriana the CRYO-EM studies. So the blue screen has hit, and I'll be happy to take any questions if you are still there.
Please unmute yourself when you want to talk. They're asleep.
You want to stop sharing the screen and be able to show people?
How's that?
Better. I don't have much to add, except as a physician who learned about actin and myosin 300 years ago, I am so impressed with how much more information, how much more detailed it is. You know, it was a ratchet when I was in medical school. Right. And now, I mean, it's wonderful to see the progress.
Yeah, it's amazing. You think that, you know, eventually there'd be nothing to study on it. But, you know, once it became clear that quite a few human diseases are caused by mutations in it, then it really kept the interest in the field. And there are a lot of accessory proteins for the sarcomere that have been discovered in the interim as well that modify myosin and actin function and so forth. So there's lots more to do.
You'll be busy.
Yes.
Now you mentioned engineering types, and I can see Parag over there, but I don't know. So
he was there. Yeah,
he's still here.
There he is. Yeah.
I'm here. I don't have questions. I mean, this is such a beautiful talk. Like, everything is really clear. All I have is like, can I please get some of your data so that we can feed it into our models and test if they Well, because you've seen the models, right, which
Yeah.
Carly made and stuff, where you have these individual rates, all of them being incorporated. And yeah, you're measuring these rates. So we like to put them in and test if our models actually work the way it really happens. So yeah.
Yeah, well, let's chat.
Yeah, I'm happy to. I think Esteban also he's here too, because he's taken over that stuff. So yeah.
I see. Okay.
Yeah.
I see Bill still in La Jolla there. That's nice. Well, as far as I'm concerned, the feat was Zoom technology. I had to come in from home because my bandwidth was so bad. It was a disaster at lab meeting this morning, so I decided I better do it here. So luckily we made it through.
Very well.
Any other questions, comments? Is not, then
actually, I might ask a question too.
okay.
So I know what you do the gliding assays for the ATPase activity. Is that even the results that you showed here, that's still what you do for the ATPase activity, or?
No, the ATPase is just a test tube assay. The gliding assays is more just to measure. I mean, they could be done simultaneously, but not quite so simple. So typically it's just a standard amount of ATP added or flooded into the system. And it's really, the gliding is just measuring the velocity of the movement.
Right.
And then the ATPase is a separate assay where, we just measure how rapidly the ATP is hydrolyzed in a test tube.
So, but with actin still has to be in there, right?
Yeah. Well, we do it both the basal form, so it's very low level, and then we add increasing amounts of actin until we get a saturation.
Okay.
But then our collaborator does the transient kinetics, where he could measure, the affinity of ADP, the off rate of the actin, and all detailed kinetics.
Yeah, I can't even imagine. The theory is so much easier.
Mm-hmm.
Well, actually, I will ask a couple more questions if it's okay. Do Drosophila have skeletal and cardiac muscle the way human beings do? Is there a distinction?
Yes, yes and no. We sometimes argue about whether should be called skeletal muscle because it doesn't have any bones, but it does have an exoskeleton, so I think that that's quite reasonable. The structure, they're both striated, but the structure is somewhat different in the heart muscle in that there are sort of circular myofibrils that go around the circumference of the heart lumen, whereas the skeletal muscles are straight in a sense. So, they're not exactly analogous to the human situation. And in fact, there are multiple types of skeletal muscle, just like there are in humans. Not really the fast and slow possibly, but there's sort of a supercontractile muscle that will contract through the Z bands, as well as the flight muscle that I've shown the structure of. So, the flight muscle does have a lot of similarities to human muscle in terms of structure.
Does the heart muscle of a Drosophila have intrinsic rate of contraction the way of a human being?
Yes, I believe that that's the case. The Adriana's there, so she could chime in if I'm wrong. But we cut the neural input, and we still get the heart myogenic beating.
Right. And so if you take the heart of a Drosophila and separate it out into cells and put it in a dish, they contract.
I believe that that's correct. That's true for the skeletal muscle as well, actually.
Actually,
yeah. That's what got me hooked in this whole field when I went to my PhD advisor's microscope 40-something years ago, and he had cultured embryonic tissue from flies and individual cells, and there were these contracting skeletal muscles on the culture dish. Yeah.
And the other thing that was just interesting, and maybe just totally anecdotal, is with your restrictive model, how the embryonic microstructure is not that distorted, but as the fly ages, it becomes distorted. And what we see clinically with idiopathic hypertrophic subaortic stenosis in human beings is it doesn't show up. I've never heard of an infantile case. I'm sure there is one somewhere, but primarily the classic place it appears is like the 18 or 19-year-old who drops dead on the basketball court.
Yes. Yes.
You know, so that it shows up in late adolescence very often as a first sign.
Right, that is definitely the case, and certainly dilated cardiomyopathy is like that as well. It's aging related. It's I guess I didn't point out this advantage of the fly is that humans, you can have a family of individuals that all have the same mutation, and one of them shows it clinically very early, and one later, and one not at all. So presumably, it could be diet, environment, but also modifier genes. So that's where the fly comes in as advantageous in that you can have a straight, clear, identical genetic background amongst all the individuals and get rid of those modifier mutations that might give you different results in different flies, or people in that case.
Very clear talk, thank you.
Thank you.
Okay, on that note, I'm gonna thank you, Sandy, for a great talk.
My pleasure.
And everybody have a great rest of the afternoon and a wonderful evening.
Yeah.
And then see you next time.
Wash your hands.
Bye-bye.
Thanks for coming, everybody.